All right, so I know it's been about a week now since we talked about the first part of cellular respiration. I'm just going to give a very brief overview and the review, but we're actually going to do an activity that'll help you reiterate what we talked about on Friday. All right, so basically we have these four metabolic pathways. All right, glycolysis, breakdown of pyruvate, citric acid cycle, and oxidative phosphorylation. All right, just as a review again, all right, we have glycolysis. We're creating two pyruvate molecules and 2 ATP is produced. Those pyruvate molecules will enter into the mitochondrial matrix and be broken down into 2 acetyl-CoA molecules. All right, so that's kind of where we're picking up today. All right, because we're going to talk about the citric acid cycle stage 3. All right, so at this point, we have acetyl-CoA, right? That's kind of the molecule that we left off on. So the citric acid cycle is a metabolic cycle where some molecules are going to enter and others are going to leave, right? But the series of organic molecules are going to be regenerated in each cycle. So it's often depicted as a circle, kind of leading into each other because the molecules are all going to be regenerated. They're not going to be used up, essentially. So the first thing that's going to happen is the acetyl is going to be removed from acetyl-CoA, and that's going to be attached to this molecule called axylacetate. All right, I actually had to write this down on how to pronounce it because I could never get it right. But axyloacetate, all right, we got it now. That is going to essentially form citrate, which is also known as citric acid, and hence where we get the namesake for this cycle. All right, so this is a series of steps that we're not going to go too in depth on. All right, so there's a series of 8 steps that essentially releases 2 carbon dioxide molecules, 1 ATP, 3 NADH, and 1 FADH2 for each acetyl group. All right, now remember we have pyruvate, so glucose is broken down into 2 pyruvate. Those 2 pyruvate are broken down into 2 acetyl-CoAs. So for each acetyl-CoA, this is what is produced. So for 2 molecules of acetyl-CoA, which comes out of 1 molecule of glucose, we would double this. All right, so the big part about this cycle is that oxalacetate is regenerated to start this cycle all over again. All right, so let's look at a diagram of this. And again, I don't expect you to know every single step, but there are some important pieces that I want you to note. All right, so first of all, we have acetyl-CoA, and we're losing the CoA group. All right, so it's just the acetyl group that is left. That is going to essentially bond and create bond with axylacetate and create citrate or citric acid. All right, so these are kind of like the 3, I guess 4 if you consider CoA a different enzyme or a different molecule that I want you to know. All right, so acetyl-CoA, the CoA leaves the acetyl group, oxalacetate forms citrate. All right, those are kind of the 4 basic molecules that I want you to know. There's a lot of other molecules in these steps, which we're not going to go too in-depth on. All right, but another big picture thing is that we are creating NADH in multiple areas of this cycle. We're also creating FADH2 and a molecule of ATP. All right, so I mentioned that oxalacetate. Oh, how many times I'm going to have to say that this time? It's regenerated, all right? So it's not used up per se, all right? And I kind of put a more specific diagram of the citric acid cycle on here. So again, you can see all those different molecules. But essentially, right before we regenerate I'm just going to say molecule O because I keep stumbling over it. Molecule O. All right, so before we regenerate this molecule, it's malate. All right, so malate is going to be oxidized, so it's going to lose its hydrogens. All right, it's going to lose those hydrogens to NAD+, so NAD+ is being reduced, and that's going to create this molecule O. All right, so that's how it's being regenerated. And so it can go through this cycle many, many times. All right, so again, I kind of mentioned this previously that One round of that cycle is one acetyl group. All right, now each molecule of glucose produces 2 acetyl groups. All right, so after 2 turns of the citric acid cycle, for 1 molecule of glucose, we get 4 CO2, 2 ATP, 6 NADHs, and 2 FADH2s. All right, so this is per 1 molecule of glucose, 2 molecules of acetyl groups. All right, so the citric acid cycle is so much more complex than we just talked about. All right, but I only expect you to know the basics of it there. There are some videos in the additional resources that I linked, which I think gives a really good in-depth explanation of some of these concepts. So if you're thinking about it, I'm like, oh, I wish I knew more about this topic. Watch those videos because they'll guide you through the whole cycle. All right, but that's just more complex than we want to get into for this class. All right, so far we've gone through 3 steps of cellular respiration. And so far we have yielded 6 carbon dioxides, 4 ATP, 10 NADHs, and 2 FADH2s. All right, so these 2 molecules on the bottom are going to become extremely important when we talk about the last step. All right, so remember, they're electron carriers. They can easily lose and accept electrons. And that's really important when we talk about oxidative phosphorylation. All right, so that's stage 4, the last stage in cellular respiration. So we have our high energy electrons removed from our electron carriers, NADH and FADH2, to make ATP. All right, so that's the ultimate result of this final step. Now this final step requires oxygen. All right, that's the reason we breathe in oxygen, is to have the oxygen for this final step here as the final electron carrier or electron acceptor. And it's a process that involves the electron transport chain. All right, so oftentimes in textbooks they won't call it oxidative phosphorylation, they'll just say it's the electron transport chain, right? Because that plays a huge part in this last step. And then finally, the last thing here is that phosphorylation is going to occur via ATP synthase. All right, so let's kind of dive into this a little bit. I do want to point out we are calling it oxidative phosphorylation, all right, because NADH and FADH2 are becoming oxidized, all right? That's where the oxidative part comes into play. Phosphorylation is occurring via these ATP synthase, which is what the later part of that phrase is coined from. All right, so often called the electron transport chain, we're calling it oxidative phosphorylation. All right, so let's take an in-depth look at this. All right, so at this step, we are in the mitochondria, but even more specifically, we are in the inner mitochondrial membrane. All right, so this is taking place across a membrane, and specifically the inner membrane of the mitochondria. Now, in this membrane, we have multiple protein complexes and small organic molecules. We can see that in this image here. You'll often find them labeled 1, 2, 3, and 4. All right. The purpose of these complexes are to accept and donate electrons in a series of redox reactions. All right, so again, this is far more complicated than what we're going to discuss, but essentially these electrons are being taken off NADH and FADH2, and they're going through redox reactions where those electrons are getting shuffled to more and more electronegative atoms. All right, the result is that it's actually going to end up being accepted by oxygen, which is going to be the most electronegative atom that it's going to encounter. So that is the final electron donor. All right, now in the process of shuffling these electrons across, we're actually setting up a gradient. All right, a proton gradient, or you can think of it as the hydrogen electrochemical gradient, often called the proton motive force. All right, we're setting this up in the course of oxidizing NADH and FADH2. All right. So essentially, we're setting up this gradient in order to provide energy for synthesizing ATP. All right, now I wanted to point out here, we have electrons, all right, in the mitochondrial matrix that are moving into the intermembrane space. We are going against a concentration gradient. All right, so when we're going against a concentration gradient, we know that that's active transport. Now in this case, we're not using ATP as the energy source. Instead, it's the movement of these electrons that are providing enough energy for these proton pumps to work. All right, so we can see that three of these protein complexes are these proton pumps: complex 1, complex 3, and complex 4. All right, they're all playing a part in setting up this gradient. All right, so we're setting up this gradient to essentially have ATP synthase happening. All right, so in our mitochondrial, inner mitochondrial membrane here, we have a lipid bilayer, right? No different than what we've seen in the plasma membrane. And it's relatively impermeable to protons, all right? Because it's impermeable to protons, right? In order to pump them out, you need those protein pumps and those protein complexes. But in order to pump them back in, we also need some sort of a pump, all right? In this case, that pump is ATP synthase, all right? And this is a pump that's essentially moving protons from the inner membrane space back into the mitochondrial matrix, but in the meantime, synthesizing ATP. All right, so they're harnessing the free energy from these electrons to synthesize ATP from ADP. The process of this is chemiosmosis. All right, so that's kind of like the big term that summarizes this all up. Chemical synthesis of ATP as a result of pushing these protons through a membrane. All right, let's take a closer look at ATP synthase. All right, so we have this electrochemical gradient set up. It's going through ATP synthase to create ATP. All right, so we're capturing the free energy as these protons are moving through. And this enzyme is essentially going to convert the energy from the proton motive force, from that electrical chemical gradient that we set up, and turn that energy into a chemical bond energy in the form of ATP. All right, so we're transferring energy from just free-flowing energy in the form of protons into energy that's now in a chemical bond in ATP. All right, so we can see ADP plus a phosphate group here. They are going to bind to this ATP synthase. Now this ATP synthase is essentially going to change conformation, so it's going to rotate. And as it rotates, it's going to create ATP. All right, so I have this diagram that's also in your book to kind of dig into this just a little bit deeper. All right, so first of all, we have ADP and a phosphate group binding to one of the subunits on ATP synthase. All right, we know a subunit is part of a protein. We're talking about a protein because we're talking about an enzyme essentially here. All right, so that's binding to that subunit. As that rotates, all right, it's essentially going to strain those bonds between ADP and the phosphate group, those bonds that are connecting them to that enzyme. And as it's straining that bond, it's essentially making them weaker so that they can break apart and form new bonds in order to create ATP. So in that last rotation of ATP synthase, ATP can be released. All right, it's a lot of information. I totally recognize this. So let's kind of sum up what we've talked about so far. And I wanted to put a review in there that NADH is a pretty important molecule in our cells. All right, oxidation of NADH is basically what makes most of the cells ATP. Now FADH2 plays a little bit of a part there, all right, but not nearly as much as NADH. There's just more NADH molecules. And that can yield up to 30 to 34 ATP molecules per one molecule of glucose. All right, so that one molecule of glucose has huge amounts of power, and then we can get up to 30 to 34 ATP. Now, this isn't an ideal situation, right? We're rarely going to get 30 to 34 ATP because the cell is going to use NADH in other processes and other pathways that it has to go through in just regular cellular functions. It's also going to utilize that proton gradient that was set up after NADH was oxidized in order to run other pathways, other purposes as well. All right, so the cell is utilizing NADH and the electrochemical gradient in multiple ways, not just in cellular respiration. So oftentimes we are not going to make that 30 to 34 ATP. All right, it's going to be a little bit lower than that because the cell is taking away some of the NADH and the gradient, proton gradient, to use in other processes. All right, so big picture review. All right, glucose goes through glycolysis. We yield 2 ATP. All right, we're breaking that down to pyruvate and then going through the citric acid cycle, producing another 2 ATP. All right, but the big payout is going to come in oxidative phosphorylation via the electron transport chain, where we get this huge payout of 30 to 34 ATP molecules. All right, so this is kind of the end all. This is where we get all our energy from, stage of cellular respiration. Now, we only talked about glucose, right? We focused mainly on glucose, but other molecules can be used for energy as well. Molecules like carbohydrates and proteins and fats, right? They can enter into glycolysis and the citric acid cycle just at different points, right? But ultimately, no matter what molecule is being used to create this energy, they're all going to utilize the same pathways, right, in order to increase efficiency. That way the cell doesn't have to have a different pathway for carbohydrates and a different pathway for proteins and a different pathway for fats. All right, they're utilizing the same pathway so we can increase efficiency. And ultimately, the result of metabolism is ATP. And that ATP is kind of the energy currency of the cell. We can use ATP to make other molecules that the cell needs. All right, so that's big picture overview. Now I have a group activity that I think is going to take a decent amount of time to finish. All right, so I'm going to pass out this activity. It's basically going to ask you to draw cellular metabolism, make sure you include certain terms, answer a couple questions. All right, so I want you all to do this individually answer them, individually draw the cycle, but you can work together as groups to make sure you're understanding this correctly. All right, I just think if everybody individually writes it themselves, it kind of ingrains it into your mind a little bit better. I have some blank pieces of paper so you can write that on. You can decide to draw this however you like, right? As long as you are showing the processes and the terms that I want you to show. All right, you can use 1 piece of paper, you can use 4 pieces of paper, however you feel necessary. If you finish this, I will give you 2 points, all right? I'm not necessarily going to grade them. I will pass them back after I've seen that you completed it. But I'm going to put the answer key up on Moodle after I've received everybody's completion. All right? So I'm going to start passing them out. No. I just, it's really boring for me to talk about this and for you just to sit there and listen about this, so we're trying to do this a little bit more interactive here. And I'm going to cycle around some sheets of paper that you can use. It's also a good time to ask questions if you're confused about any step of cellular respiration. Before you leave. Yep. So this should be done in class, turn it in before you leave. Before we get too far into it, I'm just going to say these are the additional resources. I like the 2 on the bottom. I think they were very clear and easy to understand if you need more detail. And then before Monday, I am going to have you watch a video. It's a fun video. It just kind of introduces you to fermentation. Didn't get any. All right.